I always like to get on the cutting edge and push the boundaries, and I really like to have highly miniaturized systems, very homogeneous systems. So the challenges that I still see and intend to overcome or try to help people overcome are really around immuno-oncology and looking at rapid methodologies, if you will, technologies that will allow us to study T-cell activation, CAR T types of studies in high density formats that allow us to really spread the density of the models that we have, that are very precious, right? We're indebted to the patients for giving us their tumors, but you don't always get so much you can do everything you want unless you use a highly miniaturized system. So that's what I intend to help with, try to push the boundaries in those areas in the near future. We also have technical difficulties in dispensing 3D spheres or organoids directly. So almost entirely the whole field is still disaggregating, dissociating into single cell suspensions, and then reforming 3D models. And that's something that there's a few companies out there doing a very good job, but there's definitely room for improvement and cost efficiency in doing so.
Still a lot of challenges in working with organoids. The biggest one, to my mind, is that we get a lot of variability in terms of their appearance from one organoid to the next, even from one well to its sister well. The organoid doesn't come out exactly the same. And as the organoid gets more complex and exciting to use, that variability also becomes more and more pronounced. So we're trying to find ways to guide the organoid into the same appearance every time. Another challenge that we face with organoids is that we need to find ways to deliver molecules efficiently into them and simulate the vasculature that's present in our bodies.
Yeah, I think the biggest challenge is trying to get more functionality, especially out of organoids. And that's going to take a lot of work, not only in how you generate these, the number of cells, the number of cell types, what cell types are required, but again, also with the extracellular matrix. So you're seeing these tunable matrix, the stiffness has to be controlled for functionality. So there's a lot of challenges ahead to try to make more sophisticated functioning models. And I think that's really what we're going to see. And again, how do we generate those models and have more widespread use of those models? I think it's going to be a big challenge.
For our indication, I think some of the methodologies have to be dispensing and handling of spheroids for clinical use is something that we are very involved in working out. But also for animal model studies, it's important to be able to not only synthesize these organoids in a high-scale method, but also more efficient and more reliable way of doing it. And having it being very consistent, right? Because for drug discovery, you need very consistent results tested across multiple or thousands of drug candidates. So you want to be able to make sure that your model is resilient and be able to handle that many variables.
Yeah, so organoids in 3D cultures are more complex than working with 2D cell cultures. So there's more of a learning curve for new customers and more things to think about than with traditional 2D culture. So working with hydrogels can be challenging. Staining and optimization for 3D cell cultures can be more challenging. So there's definitely some more learning that has to happen.
I think that one of the big challenges is making sure that the 3D models, especially organoids, are indeed fully functional systems. So that's very difficult to do in a dish. Another challenge is consistency. So it's very difficult to reproduce organoids in a reproducible way at scale. And so there needs to be some improvements in the methodologies and protocols that we follow to do that.